Okay, hey everybody, those of you watching this on a recording, welcome to our class meeting today. And so what we're going to do, because you have your test on Friday, or you have to take it by, I think I might push it out till Saturday. So you have to have it finished by the 11th. Okay, so you have Friday and Saturday to take it, maybe even Thursday. I don't know, it depends on how quickly I get it posted. So at least Friday and Saturday you have to take it. We'll have a regular class meeting starting with the brain, so central nervous system information, on Friday. Okay, so yeah, so that's all I wanted to say about that. So we're going to go ahead and go to our PowerPoint, because there are a few things that I want to make sure that we cover before we do our Kahoot. So after yesterday's lab, there we go. All right, so we've done all of that. We've done all of that. Okay, so, oh, we already know that. All right, so let's just make sure, let's look at the definitions of our, or the functions of our neuroglia to make sure that we have everything. So astrocytes, star shape, most abundant, forms the blood-brain barrier, soaks up the extra potassium ions that might be out in a hyperpolarized state, and some neurotransmitters. Oh, we got to watch the neurotransmitter thing too. You know what? I'm gonna, crap. I want a Kahoot, and I forgot that we were gonna do that. I'm gonna put that in as a separate video. So I'm not gonna do it live. I'm just gonna do it, and so that you can watch that on your own time. Yeah, I guess I'm gonna have to do that on a note. Okay. All right. The next thing, microglia are phagocytic, and so that they'll eat up microorganisms and any neuronal debris that might be present in the cerebrospinal fluid. Ependymal cells are going to line the cavities of the brain and spinal cord to circulate CSF. We said that. And oligodendrocytes produce myelin in the CNS. So remember, oligodendrocytes produce the myelin sheath in the central nervous system, brain and spinal cord, not on the nerves. And then there's pictures of them, satellite cells around the cell bodies within the ganglia. I don't really care that you know that, but Schwann cells, absolutely. Wrap around the axons of the neurons. One neuron has many Schwann cells, and it's in the peripheral nervous system. So make sure you remember that. Schwann cells, PNS, oligodendrocytes, CNS. All right, so what is the basic functional unit of the nervous system? Those are neurons. So the cells that can generate a nerve impulse then are going to be the basic functional unit, what's going to carry out the functions of the nervous system. So what do neurons do? They conduct nerve impulses, obviously, that's their job. Extreme longevity. Okay, how long does a neuron live? Your entire lifetime, if it's lucky. So remember that neurons, just like muscle fibers, never ever replicate once you're born. So we'll get some, now there's always some exceptions, but for the ones that are primarily in your brain and spinal cord, and then whose axons pass through the cranial nerves and the spinal nerves, those are never going to replicate during your lifetime. So if you do some activities that are going to increase the rate at which neurons die, drugs, for example, alcohol, you're going to lose more neurons than you're supposed to. Okay, so they're going to live your whole life. Some are going to die along the way. That's fine. But for the majority are going to stay, and you want to keep as many of those that you have as possible. Okay, amitotic. So this means they're not going to divide, but there are some examples. Our olfactory neurons can replicate, and then we have some in our hippocampus that can replicate as part of our memory ability. Okay, now this one's really important because your brain, of course it governs everything, but your brain is really greedy. And so your brain is going to make sure that it's getting all of the blood that it can when it wants it, with the right conditions that it wants, at the risk of other organs. And so your brain, like I said, super, super greedy. And why is it? Because it has a super high metabolic rate. It's going to be carrying out cellular respiration all the time. okay, and at some super high levels. So it's going to require constant and abundant supplies of glucose and oxygen, because that's what goes into cellular respiration. Neurons will die within 5 minutes without oxygen, but here is the issue. It doesn't mean if you held your breath for 5 minutes that your neurons would start to die, because heck no, people can hold their breath a lot longer than that. They're pearl divers, free divers, I don't know, just whoever wants to hold their breath. I mean, we can get up to 8 and 9 minutes that people can hold their breath. But here is the key. One, the heart is still pumping, so the blood is still circulating to the brain, so it's still carrying oxygen to the brain. And two, there is no obstruction in the blood vessels going to the cells of the brain. So one, if you had a heart attack and your heart stopped beating, you're going to get neuronal cell death. Number two, if you have a stroke, if you have a blockage of the blood vessels taking oxygen and glucose to your brain, you're going to get brain damage. You're going to have neuronal cell death. So that's why it's so important that we reestablish circulation as fast as possible. And remember, with a stroke, that's how you remember. FAST are your little acronym to identify as someone having a stroke. We want to get in clot busting drugs as quick as possible. We want to get that circulation going from the heart. So we want to be doing CPR to the heart. We want to defibrillate the heart, whatever we're trying to do. But we're trying to keep that circulation of blood to your brain. So remember, heart attack and stroke are going to cause your neurons to then be kept from, what's the word I want? To not be supplied with oxygen for 5 minutes. Okay, so we got to get on things right away if we want to prevent brain damage from happening. Okay, then we have the parts of the neuron, cell body, soma, whatever, is usually found within the CNS. Okay, especially motor neurons are found primarily. There's always exceptions, you guys, but 99% of the time they're going to be in the central nervous system. Remember, if we have the clusters of cell bodies out in the PNS, like we learned yesterday in lab, we have a ganglion. So remember, the cell bodies of the sensory neurons are in the ganglia out there in the dorsal root ganglion in the PNS. Okay, there's that. The processes, we have dendrites and axons, and we talked about all of that. And we talked about myelin sheath and nodes of Ranvier and saltatory conduction. And okay, so multipolar neurons. So motor neurons and interneurons structurally are multipolar neurons, many dendrites with one long axon. The cell body will be in the CNS of all the multipolar neurons, with an exception we'll learn about near the end of the semester. Bipolar in your eye and nasal cavity for smell. So they're sensory neurons. It's like they have two axons, or they have one long dendrite on one side and another long axon on the other side. And then unipolar, most sensory neurons, two axons with the cell body, or one big long axon with the cell body out to the side. And the cell bodies will be found in the dorsal root ganglion of the spinal cord, which we looked at this picture yesterday. Okay, so sensory neurons carry nerve impulses from the sensory receptors to the CNS, sometimes referred to as afferent neurons because they carry they carry signal toward the brain. Their dendrites may be the sensory receptors, or you may have some other sense cells like rods and cones in your eye. Whoopsies, sorry about that. Motor neurons carry impulses from the CNS to the effector, so they're carried, or they can be called efferent neurons because they carry the signal away from the CNS. And then interneurons, only found in the CNS, relay information up and down from the brain and spinal cord and into and out of the spinal cord and through, so sensory coming in, and then we relay to motor going out of the spinal cord. So they're always going to be in that integration in the central nervous system. We don't have interneurons anywhere else but in the CNS. And I think that's it. Okay, we came to here. So that is all for our PowerPoint, and stop sharing that. Okay, does anybody have any last minute questions before we do our little Kahoot thing? Okay, so let's Kahoot. Log in. And Kaysen, I'm glad that you finally, we finally got you here today.
I know.
All right. Okay, so here we go. And teach and classic and let's get it. There you go. Okay, so there's your code. So it's just like, I'm trying to find my speaker. Okay, so it's just like regular. So you got to go to kahoot.it.
Mrs. G, I can't see the code.
Oh, I forgot to share my screen again. You guys, come on, you gotta tell me when I'm being stupid. Forgot I wasn't sharing that. Okay, now you should be able to see it. 7346058. Yay, perfect. Because I know this works because we did it last week with 202. Oh my gosh, this morning, yeah, I had a few technical issues this morning. So it's no surprise it held over to now. We'll wait a couple more minutes. Hi, the little brain. That's so cute. I like that. Oh, I like the alien too. That's what my daughter looked like when she was born. She looked like an alien. She had really big eyes and a really big forehead. And she looked just like an alien. Velocity. Nice one. I like that. Okay. I think we'll just go ahead and get started with those people. So obviously, if you're watching this in a recording, that, you know, you won't get to play along, but you'll get to see what we do. Okay, here we go. We are ready. 62 questions. I don't know that we'll get through all of them, but we'll see. All right, so what part of the neuron normally receives the stimuli? Okay, remember the incoming stimulus comes into a dendrite. It goes out an axon, okay? So axons do not receive the incoming stimulus. A stimulus is going to be from some other neuron. It's not going to be the action potential or the graded potentials, okay? So comes into the dendrite. All right, what is the correct order of the three processes of the nervous system? Or parts, I don't know. They're not processes, but functions. Yes, sensory input, integration, motor output. So we come in, we decide what to do, we go back out. If we went sensory input to motor output, there would be no need for integration. Okay, so integration has to be in the middle. So that may be how you can remember that. All right, electrical wire is to electrical tape as peripheral neurons are to, little analogy for you. So electrical wire, so peripheral nervous system neurons would have what around them? Okay, Schwann cells, you guys, they're in the peripheral nervous system. Oligodendrocytes are in the central nervous system, and astrocytes are astrocytes are around. I know you're thinking blood-brain barrier, but they are around the dendrites, not the axons. Okay, because they're protecting the cell body. Okay, so Schwann cells in the PNS. Remember that relationship. Guarantee you that'll be a question on the exam. All right. Each neuron in our body has an estimated lifespan of yeah, very good. Our whole lifetime. Excellent. Oh, no, we didn't change positions. All right. The portion of the axon that contacts its target, like a muscle or a gland or another neuron, is the, so what part is going to come to the synapse with its target cell? I guess I should have put what portion of a neuron. Yeah, the axon terminal. Okay, cell body's not going to come in contact with the target at all, the effectors. It's going to be the axon terminal. So the very end is that part that comes in contact with the cell or another neuron or a gland, or I guess that's it. Or there aren't other things. Which of the following is not a structural type of neuron in humans? Yay, good job. There is no tripolar. Remember, unipolar, you guys, are sensory neurons. Bipolar are sensory neurons. Multipolar are interneurons and motor neurons. All right, which of the following is true concerning the relationship between structural and functional neurons? Okay, now some multipolar neurons are interneurons. All interneurons are multipolar, but not the other way around, because some multipolar neurons are motor neurons. Okay, and then yes, bipolar neurons are sensory neurons. Good. Okay, so if we flipped these around, if we said all interneurons are multipolar, then that would be right. But because some multipolar neurons are motor neurons, that's not the right answer. Okay, depolarization of the neuron refers to, what does that mean? We had this question on the last test with membrane potentials in general. So what does it mean when the neuron depolarizes? It becomes less negative, you guys. An increase in membrane potential means it's more negative. That's hyperpolarization. So depolarization is a reduction in membrane potential. Now, if I made the inside of the cell more negative, then that would be hyperpolarized as well. And you can't be both. You can't reduce and increase at the same time. You got to pick one. So it's a reduction in membrane potential. Oh, no changes. Okay. The stimulus which would generate an action potential is termed the. So what generates an action potential? It is a threshold stimulus. Good job. Okay. Here we go. All right, what is the place where a neuron interacts with its target? So another neuron or muscle or gland. So what's that place called? It is a synapse. I mean, yeah, there's a neuromuscular junction, but what is at the neuromuscular junction? It's a synapse. So it's always synapse. All right. Okay, what will opening of chloride channels in the postsynaptic membrane produce? I thought I missed a verb in that sentence. So when you open up the chloride channels in the postsynaptic membrane, what's that going to do? Or what kind of thing are you going to generate? Okay, chloride, you guys. Chloride has a negative charge. So if you are bringing more chloride channels in, because remember chloride's on the outside, if you open it up, you make it more negative on the inside. So when you make it more negative on the inside, you inhibit the neuron. It's going to be less likely to fire. If you open sodium channels, then that's excitatory. But chloride channels, that's going to be inhibitory. And that's how GABA works. All right. I guess I already gave you your points. The functional unit of a muscle fiber is, so what is going to be doing the contracting in a muscle fiber here? What is the ultimate unit? I hope I picked the right answer. I was watching, what were we watching? Infinity War, I guess, while I was doing this last night. I did pick the right one. Okay, so why is it not myofibril, you're wondering? Because what's on a myofibril that does the shortening? A sarcomere. So you got to go ultimately down to what is doing the shortening, what's generating the force. There are all the thousands of sarcomeres that are on a myofibril. Okay, now if I didn't have sarcomere as a choice, then yes, it would be myofibril. But because I had sarcomere as a choice, that's the better answer. Oh, I know, I hate those. Which is the best answer in this question? All right, which portion of a myosin molecule would be the most affected upon exposure to an ATPase enzyme inhibitor? Okay, so in other words, where is ATPase? What is ATPase? So where in a myosin molecule do we have ATPase? It is in the heads. Perfect, because that's where we get the cocking, the moving of the head so that it can be in its high energy conformation so it can do the power stroke. All right. Okay, the thin filaments are not comprised of which the following components? So which one is not part of a thin filament? That's right. Titin is actually attached to the thick filament when we're stretching a muscle so that it can return back to its resting length. All right. Okay, what is the major function of the sarcoplasmic reticulum? I don't know why I put major function. What is the function of the sarcoplasmic reticulum? Oh. Yes, it does store calcium. Nothing else is stored, you guys. And we don't want to kick the calcium out of the cell. We want to keep the calcium in the cell so that it can bind to the troponin, pull the tropomyosin out of the way. Okay, so yes, sarcoplasmic reticulum stores calcium ions. All right, what is the relationship between the T-tubules, sarcoplasmic reticulum, and sarcolemma? Yes. Okay. So the T-tubules are going to take the action potential, the depolarizing signal from the sarcolemma, because that's where the motor neuron is going to stimulate it, and carry it down to the sarcoplasmic reticulum so the calcium is released. T-tubules do not have calcium in them ever, 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 ever. They just have that sodium going in and depolarizing the membrane. All right, during a muscle contraction, sliding filament theory or model would be apparent in the sarcomere because. So how do we see it? What could we look at under the microscope and see happening? Yes, the Z discs do move closer and the H zone decreases. Okay, A bands never ever change their width at all. And the Z discs move away and the H zone gets bigger during relaxation, not during contraction. All right. Yeah, the neuromuscular junction, the muscle contraction initiation event is. So what starts the whole shebang at the neuromuscular junction? Because all of these things happen, but which one happens at the neuromuscular junction? Yes, that's exactly right. So that's the first thing that happens. The second thing that happened would be the gold one. The third thing that happened would be red. And then the fourth thing that happens is green. So like I said, all of those things happen, but the only one that happens at the neuromuscular junction is this neurotransmitter crossing the gap, binding to sarcolemma. Okay. What would the response of a muscle fiber treated with acetylcholinesterase inhibitor? So what does acetylcholinesterase do? That's right, because acetylcholine has to be removed from the receptors, and if you excuse me, kept the acetylcholine there, then you would continue to contract. So we have to get rid of the acetylcholine, and acetylcholinesterase is the thing that does that, breaks up the acetylcholine. Good. Time period between action potential initiation, production of a force in a muscle is called the so the stimulus came to the sarcoplasm or the sarcolemma, and then eventually we got actin and myosin sliding past one another. So what do we call that in between time? Perfect. It is the latent period. Good. Oh, Abby, you just kicked Camilla out of her throne. What is calcium's function during muscle contraction? Yes. Calcium binds to troponin. Okay, this is Mrs. Capulet. So she's going to grab on troponin, or little belt loops on her husband, and pull Mr. Capulet tropomyosin out of the way. All right, corpses usually exhibit rigor mortis because, so that's a cramp in a dead person. Okay, so it's not because the calcium isn't there, it's because the ATP isn't there. Because the myosin is going to bind to the actin, and then it's not going to let go. Remember, in order for a myosin to let go of an actin, another ATP has to come in. So if you're dead and you don't have any more ATP, then the muscles will stay contracted, okay, like a cramp. Oh, Camilla kicked you out again. Just barely. All right, small precise movements are controlled by what kind of motor units? Small, precise, or delicate movements. Yes, not few, because what if you had a few big ones? A few big ones are not going to let you move precisely. You have to have small motor units, okay? So really, motor units only come in two flavors, small and large. Small is for delicate movements, large is for lots of force movements, okay? So you could have few small ones, you could have many small ones. You could have few large, many large. That's not going to control your ability to move delicately or forcefully, okay? All right, muscle contraction increases in strength up to a point because because we're recruiting more and more motor units with stronger stimuli. Stronger stimuli don't inhibit motor unit activation, they recruit motor unit activation. Oh, look at MW. Came from the outside. Woo! Okay, isometric contractions come into play when an individual is. Okay, isometric. Oh, crap. I think I have two right answers on here. I do have two right answers on here. And you guys got them both. Yay. Okay. So those of you that got 4 don't get credit for it, or picked the gold one. I'm sorry, you're not going to get credit for that because I just realized, yeah, I had 2 right answers. So good on you guys. That is 100% right. Although it's not going to reflect it in the leaderboard. Sorry about that. Okay, a sprinter will use what kind of respiration to generate ATP, whereas a Tour de France cyclist will use another kind, or maybe the same. I don't know. So sprinters, what kind of respiration? And Tour de France. I'm riding for 100 miles today. I don't know. Do they have 100-mile races in a day? I don't think so. Okay, we don't know this question. All right, so sprinters. We need ATP right now. I can't wait around for my mitochondria to make it. I need it right now. So I'm going to do anaerobic respiration. Okay, that's why you breathe so heavy at the end, is because you use whatever was available to you and you made lots and lots of hydrogen ions and you got to get rid of them because you're doing anaerobic respiration. Aerobic respiration is long-term. Okay, so we're not sprinting, we're just contracting our muscles for a long time. So they'll use aerobic respiration. Okay, so these are 2 very different kinds of activities. And so this one, short-term burst of speed kind of things. This is long-term, let's keep doing it forever kind of ATP production. All right, weightlifters typically possess what kind of muscle fibers? So which ones are they trying to increase in myofibril numbers? I don't know that they typically possess. They're strengthening. That's a better way of putting it. What are they targeting? There we go. Okay, so yeah, they're trying to target their fast glycolytic because they want super strong anaerobic kinds of muscles that'll make them really big. Because, you know, if they're after bodybuilding. I guess I should have put bodybuilding. That was a bad question. But anyway, fast glycolytic is what I was after. Okay. What are the parts of a muscle fiber that actually do the contracting? Okay, so now what's doing the contracting? Yes, now it's myofibrils on that one. Yeah, because thin filaments never contract. Thick filaments never contract, you guys. They slide past one another, but they never ever shorten. They're always the same length, but myofibrils will shorten. Okay. All right, what is the period where a neuron cannot respond to another stimulus unless it is a very strong one? I should have put the other answer in there too. Very good. I should have put absolute refractory period in there, and then maybe it would have made you think. But yeah, relative refractory period. Excellent. All right. We got new people in our leaderboard. Excellent. Myelinated neurons send impulses quickly through a process called? Yeah, it's not wave summation. Okay, myelinated neurons. So we're sending a signal down the neuron. Oh my gosh, nobody got that. From node of Ranvier to node of Ranvier, in between the Schwann cells that produce myelination. So make sure you know this question. Yeah. Make sure we know that. Yeah, nobody got points on that, so that shouldn't change. Okay, when all the muscle fibers are contracted completely in a muscle, it is known as? Oh, that was only a 10-seconder. Sorry about that. It is known as tetanus, or everybody got answered in, I guess. Yeah, tetanus, not trepa. No, because they relax when we're not even contracting. We don't even have to come close to contracting all of them in trepa. Okay, so tetanus. Okay. All right, what substance stores oxygen within skeletal muscles? No, you guys. Okay, it's like hemoglobin, but it's myoglobin because it's in your muscles, stores oxygen, myoglobin. Okay, just gotta know that one. Nobody got points on that. Okay, what is a local depolarization on a neuron that can add up to a nerve impulse? Yes, those are EPSPs, baby depolarizations. Good. All right. Increasing the voltage or strength of a stimulus to a muscle produces what? No, wave summation is frequency. That's stimulating a muscle and faster and faster and faster. But if you increase the voltage or the strength of a muscle, make it heavier and heavier and heavier, then it's multiple motor unit summation. So make sure you know the difference between those two, frequency and strength or voltage. All right. Which neuroglial cell forms the blood-brain barrier? It is an astrocyte. No, microglia are phagocytic. They're going to eat up the germs, but what's going to form the blood-brain barrier? Astrocytes. All right, which muscle type would be used in an endurance activity like cross-country running or Tour de France? So marathoners and whatever, are relying on which muscle fibers to get them to the finish line. Yep, slow oxidative. Good job. Okay, most muscles contain, oh, gee, what can that be? So most muscles contain nope, it's a mixture of fiber types. Okay, so every muscle is going to have all three muscle fibers in them. So slow oxidative, fast glycolytic. However, there are muscles, those that are going to have to require a great amount of force in a short period of time, like in our legs. Okay, they're going to have some more fast glycolytic. We're going to have, depends on what part of our legs though. Okay, and then some of our muscles in our arms and chest will have more fast glycolytic, but all of them have all three. Okay. All right. Okay, the strongest muscle contractions are normally achieved by so how are we going to get the strongest muscle contractions? Yes, it is. Okay, so this isn't saying how much above threshold, but up to a maximal stimulus will definitely produce the strongest contractions. Okay. Okay, what is the purpose of myoglobin? Oh, we just did that already, so hopefully we know what it is now. Yay, we know what it is now. That's good. Okay. All right, what is a sarcomere? Yep, remember it's the area between two discs. It's not the area between intercalated discs, you guys. We haven't, those are in the heart, okay? The Z discs, though, are the edges of the sarcomere. Okay, what is the function of creatine phosphate? Covert. All right. Okay, creatine phosphate provides the phosphate, that should say, to convert ADP back to ATP. No, we never make a compound with actin. Okay, so gives the phosphate so that we can have energy for like 15 seconds, just boom. Like when the guy was pushing the rock off of himself, he used up all of his creatine phosphate. After nervous stimulation of the muscle cell has ceased, the calcium, so what does the calcium do after we don't want the muscle to contract anymore? Yes, it is pumped back into the sarcoplasmic reticulum. Yes, the level in the cytoplasm does drop, but it's because it went back in the sarcoplasmic reticulum, but nobody picked that one. All right. Okay, the striations of the skeletal muscle are produced for the most part by, so what makes it look light and dark? Exactly. Okay. The arrangement of actin-myosin. Okay, which of the following are composed of myosin? Yes, good job, the thick filament. Okay, during muscle contraction, myosin cross bridge is attached to what active sites? Oh, so the active sites on what? Definitely. Good job on the actin, myosin and actin. All right, the contractile units of skeletal muscles are, oh my gosh, how many times am I going to ask this question? About one time too many, like right now. But we should know this by now. Yay, myofibrils, exactly, are doing the contracting. Good job. All right, the main effect of TREPA in athletes as muscle contractions increase in strength is to So why are we doing TREPA and what's it, why are we getting muscle contractions increasing in strength? Yeah, we're making the calcium and enzyme actions work better. That's the warm up that's going on. It's not going to do any of those other things. And we're not going to change. All right. Okay, in an isotonic contraction, the muscle Nope. This is isometric. So the blue one is isometric. Isotonic means you're going to shorten the muscle and move the load. Okay, so we get movement in an isotonic contraction. We don't get movement in an isometric contraction. So make sure you know the difference between those two. All right. Okay, what is the muscle cell membrane called? It is the sarcolemma. All of these others are connective tissue connective tissue coverings, but the only one that's a cell membrane is the sarcolemma. All righty. Okay, what is muscle tone? Yes, it is, where some motor units are contracted and we just keep going through the motor units to keep them contracted, to keep them healthy and ready to do their job. All right, sliding filament model of contraction involves Good job. Yep, that's why it's called a sliding filament model. All righty. Okay, hang on. I got a chat question. Okay, bye. After nervous stimulation stops, ACH in the cleft is prevented from continuing stimulation of contraction by. Oh. Sorry, that there should be a space in there. No, okay. Why doesn't the ACH continue to stimulate? Because we took rid of it. Because we got rid of it. We took it away by acetylcholinesterase. Okay, so remember, we need to destroy the neurotransmitter. We've got to terminate the effect of the neurotransmitter. All right. Just about done. If a muscle were stretched to a point where thick and thin filaments no longer overlapped, then that's right. We couldn't pull anymore if they didn't overlap. Okay, good job. All right, ciliated CNS neuroglia that play a role in moving cerebrospinal fluid are, so which are the ones that have cilia on them? Ependymal cells. So that's just a matter of getting used to what the different neuroglia are. Okay, so the ones with cilia that circulate cerebrospinal fluid are ependymal cells. Alrighty. Okay, the sheath of Schwann is also called yeah. Yay, good job, you guys. That's the myelin sheath. Excellent. I like when we all get it right. Okay, excitatory neurotransmitters secreted by motor neurons innervating skeletal muscle. Okay, so it's getting the muscles to contract is what? Yep, is acetylcholine. Okay, substance released at axon terminals to cross the synapse is called, should be easy. Yep, a neurotransmitter. Nice job. Go, Sarah. Okay, a neuron that has a primary function of the job of connecting, I thought that said collecting, whose primary job is connecting other neurons is called. Yep, it's an interneuron. Good job. All right, just about done. 4 more questions. Which neuroglia control the chemical environment around neurons by taking up potassium and recapturing neurotransmitters? Yep, those are astrocytes. Okay, besides being the blood, oh, I put astrocytes twice. Okay, so yay, you 2 people over here. Why? I don't even know why I did that. Nah, we were getting to the end of the movie. Yes, astrocytes, in addition to making the blood-brain barrier, also take up potassium and neurotransmitters, removing them from the synapse. Oh, we didn't change any people. Sorry about that. Schwann cells are functionally similar to what cells? Yep, oligodendrocytes. Good job. Oop. Okay, 2 more. An inhibitory postsynaptic potential is associated with, so how is it going to be inhibitory? Yes, hyperpolarization. Nope, we're not going to open voltage gated channels. We're going to open chemically gated channels. Okay. And the last one is, what is a nerve impulse? Yep, is an action potential. Okay, friends. So that, sorry, I was lounging. Okay, so that ends our meeting information for the day. So you can review this again, watch the video, and so if you were having difficulty with some of these questions, these are very, very similar to what you're going to see on the exam. So go over that stuff. Make sure you're reading through all your notes really, really well. So I'll wish you luck with the rest of your studying for this. And then, of course, I'll see you Friday at 11. Oh, make sure Friday at 11, we're going to start CNS. So get your central nervous system notes. So chapter 12 will be the notes that you want to download for them. Otherwise, have a great day. And let's see. So I don't know when the test will open, because it depends on when I get it done, and the test will close on Saturday at 11:59. So it depends on tomorrow. I'm not planning on doing any work tomorrow because my kids are moving back from Mesa, so I gotta fix them a place to live. So I don't know. It'll be definitely open by Friday morning, so it'll be ready definitely. So at least Friday at midnight, well, 12:01 Friday morning. And oh, okay, that was that. All right, if anybody has anything else, okay, then we'll stop this and we'll get this loaded, and we'll see you on Friday.